Hello, my name is Dr. Roderick L. Roll, and today I will be talking to you about bioenergetics. Chemical reactions occur when chemical bonds are formed, rearranged, or broken. In this image below, we're looking at reactants and we're looking at products. We have an arrow that represents a yield sign. On the left side of the yield sign are the reactants, two hydrogen molecules. And on the right side of the yield sign, we have the product, H2. Below that, we have another chemical reaction that makes methane. Just understand the basic structure of a chemical reaction. On one side, you have reactants, and on one side, you have product. One type of chemical reaction is a dehydration synthesis reaction or a combination synthesis reaction. This is when you bring two smaller monomers together to form a bigger molecule. So in this example, we have amino acids being joined together to create a protein. The opposite of a synthesis reaction is a hydrolysis or a decomposition reaction. In a decomposition reaction, you're going to break a bigger molecule apart into smaller molecules. So in this example, we have AB yielding A plus B. A metabolic pathway is when a reactant yields a product. This is catabolized by an enzyme. So if we look at our image below, this is a metabolic pathway. We have reactant A will become product B. This is catalyzed by enzyme 1. Then B becomes a reactant to enzyme 2. Then enzyme 2 will convert reactant B into product C, and so on. So this is how metabolism occurs. It is a series of enzymatic reactions that are ultimately leading to products. In this image, we have a chemical reaction. We have a sugar called sucrose, that is a disaccharide. We have an enzyme called sucrase. Sucrase is going to catabolize sucrose into glucose and fructose. In this image, we're looking at the importance of enzymes. Enzymes are very unique because they will lower the energy of activation. So if we look at the first portion of the image where you see the black curve or the black line, that is without a enzyme. So think of a ball being on the left side of the hump. It is very hard or very difficult for the ball to roll up the hump. But if an enzyme lowers the height of the hump, then the reaction is more likely to occur. So this is what enzymes will do. They will lower the energy of activation. So chemical reactions occur by the use of catalysts or enzymes. A catalyst can increase the rate of the reaction without chemically being changed. In chemistry, you'll look at chemicals that act as catalysts. But in biology, we're going to look at molecules that act as catalysts. These molecules can be carbon-based. If it's carbon-based, we're going to call it an enzyme. So the enzyme is a protein that has the ability to speed up a chemical reaction. To make some of these enzymes work, you will need an inorganic component or an organic component. So if it is an inorganic component, the component is called a cofactor. A cofactor is a non-protein enzyme helper that helps to activate the enzyme. Some examples are magnesium, manganese, iron, copper, zinc, calcium, and cobalt. These are some of the trace elements that we talked about previously in Unit 1. So these cofactors must help complete the structure of the enzyme in order to turn it on. An organic cofactor is called a coenzyme. Coenzymes include vitamins. So we're going to talk about NAD, NADP, and FAD. These are derived from vitamins. In order for the enzyme to work, these coenzymes must be present. This slide shows you how an enzyme will work. The substrate will bind to the active site of an enzyme. Once bound, a phenomenon called induced fit will occur. This means the active site will tightly fit the substrate. Now you will have an enzyme substrate complex. The substrate is held in the active site by weak interactions. The substrate is then converted into a product. The product is then released. The enzyme is now available for the next reaction. Several factors influence the rate of chemical reactions. We have enzyme concentration, we have temperature, we have substrate concentration, and we have pH. We will discuss how temperature affects enzymatic reactions. Each enzyme has its optimal temperature. So based on this image, the optimal temperature for this enzyme is right around 37 degrees Celsius. Will 40 degrees work? Yes. Will 50 degrees work? Yes, but that is not the optimal temperature for this enzyme. So what this means is that as the temperature increases, the reaction rate will increase up until a certain point. Then you see that around 50 to 60 degrees, the reaction rate will decline also. Each enzyme must be in its optimal pH condition also. This pH can negatively affect enzymatic reactions. So based on this image, we have three enzymes: pepsin, amylase, and arginase. Pepsin works best at pH of 2. pH of 3, it does not work well. pH of 5, it won't work at all. So each enzyme has its optimal pH. So temperature can negatively affect enzymatic reactions, and pH can negatively affect enzymatic reactions. The third one is concentration. As substrate concentration increases, the enzymatic reaction rate will increase also. This is true until all active sites are filled with substrates. This is referred to as the saturation point. So the enzyme is without a substrate. When the substrate is added to the environment, the enzyme will bind the substrate. If you increase the substrate, more products will be formed because the enzyme is going to bind, convert, release. Bind, convert, release. This is true up until a certain point. once you have reached the saturation point, the enzyme only has one active site. So therefore, you can't produce any more product unless you increase the amount of enzyme. So if the enzyme concentration is a finite number, you will create this saturation point. In order to overcome that saturation point, you would have to add more enzyme. In this slide, it shows you that we have a low substrate concentration to the far left. Only 2 sugar molecules, but we have 5 enzymes. As we increase the sugar molecules, we're going to increase our product. But once we have too much sugar, we're not going to produce any more product than what is expected from those 5 enzymes. The fourth one is concentration of the enzyme. So as enzyme concentration increases, the enzymatic reaction rate will increase exponentially. So exponentially means it's going to keep doubling and it's going to keep doubling. So as you keep increasing the enzyme, you're going to keep increasing the amount of protein product that you can create. This slide just talks about enzyme kinetics, Michaelis-Menten kinetics, pharmacokinetics, and pharmacodynamics. This is in reference to students who want to look into pharmacy or go further into biochemistry. So let's look at how temperature and pH can affect the structure of these proteins. So remember, an enzyme is a protein. So enzyme denaturing occurs when the protein unravels from its native shape. Disruption of hydrogen bonds, ionic bonds, and other weak interactions has occurred. Remember, we talked about fibrous protein and globular protein. In order for a globular protein to work, it must be in its tertiary configuration, at least. What holds the tertiary structure together are these bonds, the hydrogen bonds, the ionic bonds. If the temperature is elevated too high, it can disrupt the hydrogen bonding interaction or the ionic bonding interaction. This will make the globular protein unravel from its 3-dimensional shape to a 2D shape or a primary structure. This means the enzyme will no longer work. So if salt, pH, temperature, or any other aspect of its normal environment is changed, the weak bond holding the native shape will be severed. Therefore, it will not work. This slide here shows the optimal temperature for most human enzymes. It's right around 37 degrees Celsius. Also in this image, it shows you a very unique bacteria. This bacteria enzymes can work at 77 degrees Celsius. This slide shows that the optimal pH for pepsin, which is an enzyme that's in the stomach, is right around 2, versus the optimal pH for trypsin, which is in the intestines, is pH of 8. Let's look at enzyme inhibition. Enzymes will normally bind a substrate in the active site. But sometimes a drug or another molecule can actually bind to that same active site. If it can bind to that active site, then it is competing for that active site. We're going to call that competitive inhibition. Now let's look at C. If the drug or molecule binds to a different site and changes the active site's configuration so that now the active site will no longer fit the substrate, then that is non-competitive inhibition. So when a mimic competes for the active site, it can reduce product formation. This is called competitive inhibition. In our image below, we have a substrate and an inhibitor. They both have the same geometry. They have the same shape. When the inhibitor binds, for that split second, a product will not form. Then, when it is removed, there's a 50% chance the substrate will bind. Then when the substrate removes, there's a 50% chance another inhibitor will bind, and vice versa. So when the drug or the inhibitor is present, it will reduce the amount of product that will be performed, that will be produced. This can be overcome by increasing the substrate concentration. So if there's only a 50% chance to create the product because you have drug present, then just increase the substrate. If you increase the substrate, it's going to reduce the value of 50% maybe down to 45, maybe down to 40, maybe down to 30, maybe down to 10%, depending on how much of the substrate you reintroduce into the environment with the enzyme. So to overcome inhibition, competitive inhibition, you must increase the substrate. When a molecule does not directly compete for the active site, but it binds to a different site, that is called non-competitive inhibition. Let's look at our picture below. We have our same substrate. Our same substrate wants to bind to its active site, but the inhibitor binds to a different site that is called an allosteric site. This is going to change the three-dimensional structure of that active site. Now it negatively affects the production of the product. This is non-competitive inhibition. Enzyme inhibition can be separated into reversible inhibition and irreversible inhibition. Irreversible inhibition, once it is activated, it can't be stopped. Versus reversible inhibition, once it occurs, it can be stopped. Reversible inhibition is separated into competitive inhibition, uncompetitive inhibition, and non-competitive inhibition. Non-competitive inhibition is when the binding has occurred from the substrate to the enzyme, but this other molecule can bind also, and it will reduce the product that is formed. The uncompetitive inhibition is when the substrate and enzyme has bound to one another. They have formed the enzyme substrate complex. Then this third molecule can still bind to the ES complex. This is uncompetitive inhibition. And then finally, competitive inhibition. That's the one we'll spend a little more time talking about. The competitive inhibition is when the molecule competes for that active site. This is competitive inhibition. The inhibitor binds to the same site as the substrate, but this is reversible because the inhibitor can be removed and the substrate can bind. An example of reversible inhibition is with the drug naloxone. Naloxone is an opiate antagonistic drug. This drug is a competitive antagonist that binds to the opiate receptor with higher affinity than the opiate itself. This is an example of reversible inhibition. Irreversible inhibition is typically seen with toxins and poisons. An example is with sarin gas and nerve gas. Once this binds, the substrate cannot remove the inhibitor.